Good morning. Good afternoon, I guess. That clock's a couple minutes slow, so it really is afternoon. Happy Wednesday. I was a little behind in getting some of the videos done. I have now done them with the exception of Mondays. Monday there was a technical problem with the video. There is no video of Monday's class. There is audio, and I've posted that, but every now and then we have a problem, and the problem was my fault, actually. I pushed the little record button on the camera, but then I did something else, and I didn't realize that doing something else took the record off, so there was no video, unfortunately. So I apologize for that. Hopefully you took good notes, and hopefully the audio will help you with that. And yes, I did double and triple check today that the recording is going, so this is good. All right, so we're moving along. I'm going to finish up talking about characterization of proteins today, and move on to a topic that I think most students find very interesting, and I think is personally one of the most interesting proteins that's in biology, and that's hemoglobin. So we'll be talking about that most of the day today, although I've got a few things to finish up with on protein characterization. One of those things I want to finish up with is describing a technique that we use in the laboratory quite frequently to basically tag and follow a protein. Okay? And this technique is called Western blotting. How many people here have run Western blots? Probably, yeah. It's becoming a fairly common laboratory technique. Western blotting is a way of not only separating proteins by a gel, but then using an antibody to identify which of those proteins on the gel is your protein of interest. And so I want to say a little bit about how that works. Then I'll say a couple things about spectroscopy very briefly, and we will move on to hemoglobin. So Western blotting is something that we need to understand, I think. And Western blotting relies upon two things. One, separation of proteins by SDS page. So we talked about that last time, how we use SDS page to separate the proteins on a blot. And the second is using an antibody to bind to that tells us where our protein of interest is. Now, so to understand that, we need to understand a little bit about antibodies. Antibodies we'll talk about next term, and I'm sure you've had classes where you know what they are, but I will briefly say a couple words about them. Antibodies are, of course, proteins of your immune system, and these proteins of your immune system function by recognizing and binding to very specific structures. Okay? So they recognize and bind to very specific structures. So the way that they work in your immune system is you get vaccinated, for example, against smallpox. What that smallpox vaccine has in it is some proteins, and some protein structures from the smallpox. Actually, smallpox is not a good example, but measles, for example. Some proteins that are from the measles virus, and your immune system recognizes those proteins as foreign. And the immune system then does what's called mounts a response, and it makes millions and billions of antibodies that all recognize and bind to those proteins. So that when a real measles virus comes along with its own proteins in there, the immune system attacks it before the virus has a chance to get a foothold and so forth. So that's how your immune system protects you. In the laboratory, we isolate those antibodies that bind to specific structures and use them in a way I'm going to describe to you here. The antibodies that we use in the laboratory typically come from a couple of different organisms. Bunny rabbits are very popular. The process of getting antibodies from bunny rabbits doesn't kill them, doesn't hurt them. You take a little bit of blood from them, and that's really about the only component of the antibody isolation that has any effect on the bunny rabbit. The way you do this in the laboratory is you have a protein that's of interest to you. You inject it into the bunny rabbit. The bunny rabbit's immune system makes a lot of antibodies against that particular protein, and then you take blood from the bunny rabbit and isolate those antibodies that you can then use. So when you've done that, you have a collection of antibodies that bind to this protein that's of interest to you. All right? So now if you understand that, what I want to describe to you is how we use those antibodies to in the laboratory in a Western blot. So let's say, for example, I am working with a cancer cell, and a line of cancer cells, and I am interested in a particular protein in there. This protein might be one that I had identified on a 2D gel electrophoresis and saw, whoa, this guy is being made in this cancer over here. I would really like to know what it's doing in my cell line and so forth. Okay? So what I would do is I would take that cancer cell the cancer line that I'm working with, grow up some cells, bust them open, and instead of doing a 2D gel or anything like that, which is a pretty elaborate technique, I would just take that mixture of proteins and I would load it onto an SDS page gel. So what I would have in that lane of my SDS page gel would be a mixture of all of the proteins that were present in that cancer cell. If I were to look at it after I ran the gel, I would see band, 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 all kinds of bands. And I say, whoa, well, that's a lot of bands there. I think that one's mine, but I don't know if that one's mine. Maybe it's another protein that's the same size that the body makes a lot of, for example. I don't really know. So I'd like to know, is my protein present in this mixture of bands that I see on this SDS page gel? So to do that analysis and to answer that question, I do what's called a Western blot. So Western blot involves the following steps that I'm showing you here. First, we do the SDS page that I described, and that gives us this thing that we see on the left side. Next, I take that SDS page gel and I lay it on a membrane. Okay? That membrane is a special type of paper that is designed to bind to proteins. And I actually use an electrical current to transfer those proteins out of the gel and onto that piece of membrane paper. So when I'm done, I have a piece of paper that was the same size as my gel, and it has lined up with it all of the proteins that were on that gel, and they're all stuck on that paper in the same location that they were on the original gel. I then take that membrane and I put it into a little plastic bag that's got some buffer in it. And I do various things to block it so I don't get random binding and so forth, but that's not really critical for our purposes. What is critical for our purposes is that I add to that mixture the antibody that will bind to that protein of interest. And the antibody that I have modified in some way so that it in some way either gives me a color or light or something. And we don't need to worry about the technical details of that. So that antibody has some sort of a color or an enzymatic reaction or something associated with it that I can later use to tell me where that antibody is present on that membrane. So I let it sit in this solution for a while. I pour off the solution. I take the membrane. I rinse it off and wash off all of the free antibody. That is the antibody that isn't bound to anything, but that just happens to be laying there on the paper. And I do my technique to expose that color or chemical reaction or whatever. So usually it involves a color of some sort. And when I see that color, I know the location of that antibody. And now, by knowing the location of the antibody, I know the location of the protein that I was interested in studying. So I would see this and I'd say, yes, that protein that I was interested in is present in this mixture from this cell. And here it is. And if I know the molecular weight from a ladder or something like that, I can determine the approximate molecular weight and so forth and understand that. So that's what's involved in Western blotting. Western blotting is a very powerful technique because it's very simple and it tells us if a protein is present or not, and it gives us a little bit of quantitative information. Western blots aren't real good in terms of quantitative information, but they give us a little bit of quantitative information so that we can see, in fact, is the protein that we're interested in present on this gel. Yes? Well, you isolate the antibody of interest first. So you've got a protein that you're interested in. You inject it into a bunny rabbit, and the bunny rabbit makes antibodies against that protein. So you isolate those specific antibodies, and now they're only going to bind to the protein, or hopefully only going to bind to the protein that you're interested in. Make sense? Other questions? Okay, so that's Western blotting, very useful technique in the laboratory. The last two things I want to talk about are a very brief discussion of what's called spectroscopy. And spectroscopy uses electromagnetic radiation as a means of studying molecular structure. These are incredibly powerful and incredibly intricate techniques. Okay, since I'm going to spend about maybe 5 minutes talking about them, you can imagine we're not going to cover them in a lot of depth. Okay? But I think you should understand what they do if you don't understand how they work. The first technique is called Xray Crystallography. And Xray Crystallography is a technique that allows us to determine molecular structure quite precisely. To do X-ray crystallography, one has to have a protein, or it could be a nucleic acid as well, or some other molecule, but usually it's a protein, that one has purified essentially completely. So that those purification processes that I talked about, I said we really wanted to get it as pure as we possibly could. This is really, really important for this technique. Because in X-ray crystallography, what one is doing is one is making a crystal of that protein. And to make a crystal that's pure and that is useful, it has to be apart from everything else. Well, how does one make a crystal? The process of making a crystal is simple in theory, but extremely finicky in practice. In essence, it involves taking a protein or molecule of interest, making it in high concentration in a solution. Okay? And then over time, very slowly letting that solution evaporate. And as the solution evaporates, the solubility of that molecule in the solution decreases and it starts to form a crystal. And what a crystal is a regular arrangement, orderly packing of the molecules. That's what actually makes a crystal. So for X-ray diffraction purposes, this crystal has to be very precise and very regular in its repeats of the protein within. And that's where some of the issues with making crystals forms. But if you make a good crystal, you can take that crystal and you can insert it into a machine called an X-ray crystallography machine, great name, which will in turn use an X-ray source to shoot X-rays through that crystal. And this X-ray is shown as if it comes from one place, but in fact it comes from all angles in essentially 3 dimensions. What happens as those X-rays pass through the crystal is they interact with the electron clouds of the individual nuclei, and they get diffracted, that is, they get bent. The bending of the X-rays as a result of that can be traced back to the original source, which is in the crystal, and using some very sophisticated techniques, one can determine in that crystal the location of every atom in it. It's a phenomenal process. It's a very time-consuming, it's a very elaborate process, and again, we're not going to go through how we do it. But the process allows us to determine the 3-dimensional position of hundreds of thousands of atoms in a very, very large molecule. A very, very amazing thing. Okay? So X-ray crystallography is a very, very powerful technique for that purpose. When I know the 3-dimensional structure of a protein, what I can do is I can use the knowledge of that to design molecules that may interact with that protein and perhaps inhibit its function. That's at the core of modern drug design. Yes, Cassandra?
Does it just tell you the structure, or does it tell you what atoms are actually in?
It tells you the structure and the atoms. So you know where everything's at in there. You know where the alpha helices are. You know how far, you know how many angstroms this carbon is from this hydrogen, etc. You know 3-dimensional information. I mean, it's an enormous amount of data that's contained in a single structure.
So you even know the elements that are in it?
You know the elements that are in it. That's correct. Yeah. So you know the atoms as well as their location. Now, as I said, that's sort of at the forefront of modern drug design. This is when people say, what is biophysics? This is one of the things that biophysics is. Biophysics is structural biology, understanding the relationship between structure and function. And the people who do this type of analysis are all biophysicists. In my department, Dr. Andy Karplus is a world expert in X-ray crystallography and uses it extensively in his research. Now, X-ray crystallography has some great advantages. It has some disadvantages. Some of the disadvantages, you have to make crystals. And making crystals, as I hope I've communicated to you, is not trivial. And the second is that crystals aren't necessarily the structure that we see in solution in cells. We think they are, and they're probably not far off of that, But we're really interested in the dynamic structure of a protein as it's dissolved in water, not in a crystal. We have to look at other techniques. And so another technique for studying molecular structure that takes a very different approach is called nuclear magnetic resonance. Nuclear magnetic resonance is based on the fact that certain atomic nuclei have a spin associated with them. A spin. That spin is affected by magnetic fields. And by shooting radio frequency waves through there, we can actually flip the spin of nuclei from one state to another state. And that's, you've had organic chemistry. I'm not going to go into it in any more depth than that. But suffice it to say that the spin of individual nuclei varies with the environment in which they find themselves. Okay? What did I do? Put the same thing, didn't I? So we see the spin changing as a result of electromagnetic radiation interacting with that nuclei. And the frequency and strength that it takes to do that is determined by the molecular structure. Let me just show you an example. Okay? So here is a nuclear magnetic resonance spectroscopy plot in terms of chemical shift, and I'll explain that in a second, for a very simple molecule. Okay? This is your Thirsty Thursday molecule. All right? Now, this guy here has basically three types of hydrogen in it. It has methyl hydrogens, it has methylene hydrogens, and it has a hydroxyl hydrogen. Okay? Each of these is affected by that electromagnetic radiation differently based on the environment, because they're in different environments. This guy in the middle is close to each of these protons on the end, and we're looking at protons here, by the way. This proton is only close to these two, and this one is only close to these two. So it turns out that they have different ways of responding with that electromagnetic radiation. That ways of responding is measured as a chemical shift. And that shift results by comparison to a standard. And again, we're not going to worry about that. The important thing is that using nuclear magnetic resonance, I can do two things. Okay? I can determine the different types of protons that I have present in a molecule, okay, which tells me an awful lot about those molecules. And I can tell something about how they are arranged in space using other types of nuclear magnetic resonance information. The beauty of this is I can do it in water. I don't have to make a crystal. And I can do it in an environment that's very similar to what the protein or nucleic acid would be present in cells. Now, it's way beyond what we're going to talk about here to understand how that is put together to determine molecular structure. Okay? There is a plot of what's called 2D NMR, which I'm not even going to bother to go through at the moment. But using this information, one can also determine the position in space of nuclei, both the composition as well as their location in three-dimensional space. NMR has some limitations that Xray crystallography doesn't have. The bigger the molecule, the much more complicated it gets for this. And so Xray crystallography will have a wider range of molecules that it can work on. But nonetheless, NMR is very, very powerful for working with aqueous solutions of molecules. Okay. Questions about that? Pretty simple analysis. Simple analysis of a complicated topic. All right. Well, that finishes what I want to say about protein characterization. Now I want to move to something that's much more biological and something that I hope that you will find to be of considerably more interest. And this is hemoglobin. With hemoglobin, we're going to start now taking the knowledge that we've gained about structure, about protein structure, about ways of analyzing protein structure, apply it to a protein, and understand what that protein is doing in the body and how it does it. And I hope to convince you that hemoglobin is almost magical. Okay, so hemoglobin, as everybody knows, is the protein in our blood that is responsible for carrying oxygen. I contrasted hemoglobin with myoglobin earlier. I said myoglobin is a protein that's very good at storing oxygen. We'll see why that's the case in just a little bit. Okay? So hemoglobin is great at carrying oxygen. Myoglobin is great at storing oxygen. They're actually related. You notice they both have the word globin in them. Myoglobin, hemoglobin. They differ, however, in their quaternary structure, for example. Hemoglobin has 4 subunits. It has quaternary structure. Myoglobin only has 1 subunit and therefore has no quaternary structure. The 1 subunit of myoglobin is related to the 4 subunits that we see in hemoglobin. Both the myoglobin subunit and the subunits that we see in hemoglobin contain a heme group. And a heme group is where the oxygen is actually held during transport or storage. Okay? Myoglobin, I'll remind you, looks like this. You probably could look at that and say, oh yeah, myoglobin, right? And hemoglobin, the heme group that's within this molecule looks like this. The heme group is a good example of something we call a coenzyme, or it's not really an enzyme, but a cofactor that is a component of a protein that's not an amino acid. When we talk about a protein, we think of amino acids. This clearly is not an amino acid. It's an extra group that's been added to this protein called globin. It has a ring structure that you see like this, and in the middle of this ring structure, there's an atom of iron. And of course, I think we all know that we need iron for having a good functional hemoglobin. This iron is in the ferrous state, meaning it's iron plus 2. And only the iron plus 2 will work in hemoglobin, or myoglobin for that matter. Okay? So both the hemes of the 2 different globins, myoglobin and hemoglobin, are the same. They both use an iron with a plus 2. If you oxidize this iron, which you can fairly easily do by exposing it to molecular oxygen out in the, you take blood, you leave it out here, what will happen is the iron that's in there will become oxidized from the plus 2 state to the plus 3 state. It doesn't happen in the body. Okay? That's kind of cool. That's kind of good because you don't want to oxidize all your iron in there. You're not going to carry much oxygen if you do. One of the reasons when they take blood the way they do is they're reducing the exposure of that blood to oxygen. You notice it comes in little bags, and the little bags don't have any air in them. That's why. It's one of the reasons why. It keeps it from becoming oxidized and damaged. Okay. The heme group is also known as a protoporphyrin ring. Is just another name for this. And this ring, we won't talk about it again, but I will point out to you that for anybody who's interested in botany, that this ring is almost identical to the ring that's found in chlorophyll. The main difference in chlorophyll is that the iron is replaced by a magnesium. And chlorophyll, you know, of course, is this complex that's involved in capturing energy from the sun. Okay. Well, we know that hemoglobin and myoglobin bind to oxygen. All right? What I'm going to show you first is what happens, and most of the figures I have here relate to hemoglobin. The same thing happens in the binding of oxygen in myoglobin, okay, within a given subunit. All right? The difference between hemoglobin and myoglobin in terms of function is what happens between the subunits. That's what we're going to see. But the binding itself, which happens within one subunit, is the same in myoglobin and hemoglobin. All right, so what happens? Well, here's that protoporphyrin ring system that was of this nice flat ring structure that we saw, and hemoglobin was in the middle of it. We're looking at it from the side. And from the side, we can look at this and we notice that that iron atom is hanging just a little tiny bit below the plane of the ring. It's held on 4 sides in the ring, and it's held under the ring by a histidine. Okay? It's held under the ring by a histidine. That's very important. You say, what's the histidine attached to? Well, it's attached to the rest of the polypeptide chain. So it's all a part of that protein. The histidine is part of that globin protein, whether it's myoglobin or hemoglobin. Okay? What you see on the left is the heme group in the absence of oxygen. What you see on the right is the structure of that whole complex in the presence of oxygen. And what you notice is that happens is a very, very, very subtle change. a movement of a fraction of an angstrom, this oxygen, when it comes in, it grabs a hold of that iron and it pulls it up. It literally lifts it up. All of the properties I'm going to describe to you about hemoglobin arise from this pulling up. Okay? And I hope I impress with you, on you with this, what I'm going to say, that very, very small changes in the structure of a protein have enormous changes on the action of that protein. All right, so for the next few minutes, I'm going to talk only about hemoglobin. Okay? I'm not going to talk about myoglobin. Both of them have the ring. Both of them get pulled up. But in hemoglobin, an additional thing happens. All right? When this guy gets pulled up, okay, this histidine, I noted, is attached to the rest of the polypeptide chain, which means that next to histidine, there's another amino acid down here. If I pull histidine up a fraction of an angstrom, what do you suppose is going to happen to the amino acid that's attached to the histidine? It, too, is going to get moved. And yes, that happens in myoglobin, too. Okay? And the amino acid that's attached to the next amino acid gets pulled or changed in some slight way. And each of these changes is, in turn, slightly changing the chemical environment in which those amino acids reside. This very slight structural change inside of this hemoglobin, or in this case, myoglobin as well, causes the overall three-dimensional structure of this entire subunit to change very slightly. Okay? So I like to think of this as there's the foot bone, which is attached to the shin bone, which is attached to the knee bone, and you pull on the toe, and eventually you're going to pull and you're going to change the structure of the organism that's attached to it. Okay? In this case, the organism is a molecule. It's a molecule of a globin molecule. So by pulling on this thing on the end, which is actually in the middle of the molecule, I am, in fact, changing the overall three-dimensional shape of that unit very slightly. Now, the hemoglobin part, the part that's unique to hemoglobin is this. Hemoglobin has four subunits, two identical alpha subunits and two identical beta subunits. The alphas are like the betas, and the betas are like the ones in myoglobin. So they're all very related proteins. Okay? What I've just described to you could have happened in an alpha subunit. It could have happened in a beta subunit. It doesn't matter. They have the same net effect. But I've had a hemoglobin that's got four subunits. It's got no oxygen in it, and the first subunit has bound to an oxygen. That first subunit has changed its structure very slightly, and in the process of changing its structure very slightly, its interaction with the other three subunits changed. That, in turn, causes those subunits to change very slightly in structure. And that very slight change in structure that they have makes them much more receptive to binding oxygen. So to say it in another way, the binding of the first oxygen to one of the subunits changes the other subunits so that they are much more willing to bind oxygen than the first one was. The second subunit will bind oxygen, and it too will get changed. The third subunit will bind oxygen, it too will get changed, and then the fourth subunit. So each time you bind oxygen, that big molecule becomes much more receptive to binding additional oxygen. Each heme group binds one O2. So in a four-subunit molecule like hemoglobin, there are four O2 molecules that get bound. Now, this turns out to be very, very, very critical for hemoglobin's function. Myoglobin doesn't have this happen because once it's bound one, there's no subunit for it to interact with. It can't change the binding of another one. Hemoglobin exhibits a property that we call cooperativity. And cooperativity we're going to see examples of. Cooperativity occurs when the binding of one molecule to a protein changes the binding affinity of the other subunits of the same protein. Binding of one oxygen increases the affinity of the other subunits for additional oxygens. Okay, so before I go on, I want to make sure that concept is clear to you. Do you understand what cooperativity is? Yes, Laurie.
As each oxygen binds, does the affinity go up with each oxygen?
It goes up with each one, yes. Okay?
Is that where it says in the second one where there's now a carbon bound and a nitrogen bound? Is that correct?
Say what now?
And then in this diagram on the
Uh-huh.
right, there's a carbon atom over on the
Where?
no outside of the ring on the right structure.
This?
Yeah, that.
Those are hydrogens.
Yeah, but over there.
Oh, they've miscolored it. Very good eyes. That's right. It's miscolored. Yes, that's a hydrogen. That's not a carbon. It doesn't change from a hydrogen to a carbon by binding. Good eyes. Very good eyes. I never noticed that before myself. When I was, I helped to author the third edition of a textbook several years ago. And I wasn't involved in the first two editions, and so I was working through with the authors on the third edition. And I looked at this figure that had been in the first two editions. It had gone through probably hundreds of eyes. And I looked at it and I said, that carbon's got 5 bonds. And it did. They said, how did we not see that? It was kind of odd. So it takes a different set of eyes sometimes to see something that you don't otherwise see. Okay. We don't need to worry about that for the moment. Don't need to worry about that for the moment. All right? But now I want to talk about what's happening in hemoglobin. This is a representation of the structure of hemoglobin, both in ribbon form and in molecular model packing form, as you see over here. Though it looks different, the basic structure is the same. What happens in this structural change in hemoglobin, okay, is that you see this little pocket here in the middle, and you see a little bit of it right here. One of the things that happens in going from hemoglobin that has no oxygen to hemoglobin that has oxygen is the shape of this pocket changes very slightly. And we're going to see this change in this pocket is very important because it turns out that there's a molecule that can fit in there only when there's no oxygen present. Okay? Now, another thing to know. We refer to these different shapes of these subunits that I've described as having 2 forms. Okay? They're called T and R. Okay? The T form of a protein T stands for tight or taut, meaning that it's not very flexible and it generally doesn't have very strong binding affinity for molecules. The R state, R stands for relaxed, it's not tight. The R state of a protein refers to its, and by the way, the T and R refer to protein structures, not I said molecule, but it refers to protein structures. All right? The R state of a protein refers to the relaxed state, and that state has a high affinity for binding. So what's happening when these proteins change shape is that they are changing essentially from T to R. They are changing shape, and those changes in shape result in changes in this pocket, which we're going to see are very, very important for hemoglobin. Okay. Let's look at some graphs, okay? Now, with graphs, as I said before, it's important to understand the axes, understand what's being plotted. We see in this plot that we are measuring the fractional saturation, meaning what percentage or what fraction of all the myoglobin molecules are saturated with oxygen. 1 being 100%, 0.5 being 50%, 0 being 0. If I had something up here, I have all of the molecules being saturated. Something down here, I have none of the molecules saturated with oxygen. Okay? What I see on the x-axis is the partial pressure. That is the pressure of oxygen, or the amount of oxygen that's present. What we see when we look at hemoglobin is that it takes a very tiny amount of oxygen for this guy to get up to 50% saturated. What that tells us is that myoglobin has very strong affinity for oxygen. It sees oxygen, it loves to bind to it. And that's really good quality for a storage protein. In your muscles, myoglobin functions as a sort of a reservoir for oxygen. It's sitting there holding on to oxygen. Under most conditions in the muscle, The oxygen concentration is over here, and when it's over here, we see that myoglobin is pretty well saturated, or pretty close to saturated with oxygen, meaning all the molecules are holding on to oxygen. Only when you're really exerting and you're really getting your oxygen concentration in your muscles very low does it fall to this level, and when it falls to this level, myoglobin starts letting go of its oxygen and giving it to the muscle cells that are starving for oxygen. That's very good. I like to compare it to the uninterruptible power source that you may have on your computer. Okay? That's there to power your computer if the electricity goes off so that you can shut down and save files, right? It doesn't come on until the electricity goes off. This doesn't start releasing oxygen until essentially the cells start running out of oxygen. But when it's there, this keeps a supply of oxygen going to those muscle cells for a while. You might wonder, well, when does it happen? How does that happen? It happens because your muscles can use oxygen faster than your blood supply can deliver it. When you get exerting very heavily, you're using oxygen faster than it can be delivered. Having a backup supply of oxygen in myoglobin makes a lot of sense. Okay? Now, because myoglobin has strong affinity for oxygen, it's not real good at transporting oxygen. If you use myoglobin to carry oxygen in your body, you'd go to the lungs and grab all this oxygen. That would be great. But it wouldn't give it up unless your body were absolutely starving to death. You don't want to have that in a transport protein. You want to have a transport protein that has flexibility. You want it to have high affinity for oxygen when it has a lot of oxygen present, and you want it to have low affinity for oxygen when there's very little oxygen present. And that's exactly what we see with hemoglobin. Now, here's the same plot. We notice that with hemoglobin, if we compare the affinity that is the fraction of hemoglobin that's saturated with oxygen at any given concentration, over here, okay, we're still at 50%. What percentage of hemoglobin has oxygen? Essentially 0. If we go all the way out here, which is more in the area where muscles are actually doing exertion, only maybe a third of the hemoglobin molecules are holding on to their oxygen. They've given it up. When the hemoglobin gets to the lungs, where the oxygen concentration is up here, look what the affinity for oxygen is. Great. Hemoglobin grabs onto that oxygen. It's got great affinity when the oxygen concentration is high. It's got low affinity when the oxygen concentration is low, which means it grabs oxygen in your lungs. It lets go of it when it gets out to the tissues. Very, very important quality. This cooperativity is what gives rise to this, because what we're seeing is hemoglobin changing its affinity based on the oxygen concentration. Its affinity is changing as the oxygen concentration changes, and that happens because of cooperativity. Now, just as we imagine that when we bind that first oxygen molecule on there and we have greater affinity for the second, the third, and the fourth, so too when we get to the tissues and that first one jumps off, the next one's more likely to jump off, the next one's more likely to jump off, and the next one's more likely to jump off. Pretty cool. So cooperativity helps the body, that is, helps hemoglobin. Please turn your cell phones off, folks. All right? Helps hemoglobin to serve the oxygen needs of the body. All right. This figure shows, it's a little bit more complicated, but it shows the approximate concentration of oxygen in the tissues. Okay? So way up here we see about 80 or 80+% of the myoglobin is still holding on to oxygen at a point where hemoglobin is holding on to maybe 30% or so of its oxygen. All right? At the same oxygen concentration, hemoglobin is giving up oxygen, myoglobin is holding on to it. We get up here at high oxygen concentration, and they both have about 100%. Here's the concentration in the lungs right here. Okay? And in the lungs, we see they both have, the concentration we have in the lungs, they both have essentially 100%. Okay, so that really helps the body to have oxygen in the places where it needs it. All right? Blah, blah. If we exercise, we lower that level, et cetera, compared to rest, and that's not a surprise. Okay. Now, I told you that there were quaternary effects, that is, structural effects between the four subunits when hemoglobin binds oxygen. Here's deoxyhemoglobin, meaning it has no oxygen. Here's oxyhemoglobin, which has 4 oxygens. Notice how this inner pocket has changed in shape. And this inner pocket was where I told you that a molecule can bind. All right? Now, the molecule that can bind in this inner pocket, I'm just going to name it for you right now. We're not going to, the structure isn't that important. It just fits in this place. It's called 2,3-BPG. 2,3-BPG. All right? Has a longer name, stands for 2,3-bisphosphoglycerate, and we'll say more about it later in the term. But for right now, 2,3-BPG is all we really need to worry about. 2,3-BPG is produced by cells that are actively respiring. Muscle cells that are doing a lot of work are producing 2,3-BPG. 2,3-BPG, when it binds to hemoglobin, favors the T state, the state on the left. 2,3-BPG, when it binds to hemoglobin, favors the unloading of oxygen from hemoglobin. Because what it does is it stabilizes this deoxy state. It stabilizes that. And the binding of it, during the binding, oxygen is being dumped off of there so it can get into this state. 2,3-BPG has a very important biological function there. When we look at actively respiring tissues, they're producing 2,3-BPG. We'll discuss later in the term why they're doing that. But they are producing it, and they also happen to be the cells that need the most oxygen. That's very good. So it's being produced at the location where oxygen is being needed. When it binds to hemoglobin, it favors release of oxygen. The cells are getting what they need. That's very good. Yes, Lenny.
Out of the molecule as well?
Is it coming?
2,3-BPG come out of the molecule as well?
The 2,3-BPG is, it can come in and out, yes. And that'll be important for what I'm going to say in just a bit. Okay? So 2,3-BPG can cut, her question was, can 2,3-BPG come out of the molecule as well? And it can. It's not, there are no bindings that we're going to talk about here that are covalent. They are always on-off. Okay? Now, so the point I hope to make is that 2,3-BPG is made by actively respiring cells. It favors the state on the left, which favors the dumping of oxygen by hemoglobin. The cells are getting the oxygen that they need. Very, very good thing. Okay? Now, that's how 2,3-BPG functions normally. Now, smokers, people who smoke, have 2,3-BPG in their bloodstream at higher concentration than non-smokers. Oh, what does that mean? Well, Laney's question was relevant here. 2,3-BPG comes on, comes off. So as the hemoglobin is traveling in the bloodstream, traveling through the body, 2,3-BPG is coming on, coming off, coming on, coming off. And it turns out 2,3-BPG can be, in most cases, fairly well broken down by the body. So though it binds to hemoglobin at the place where the tissues are, by the time it makes it all the way back to the lungs, it's come on, it's come off. It may have gotten grabbed by something else, and it got broken down. The hemoglobin is not being left in the T state. The hemoglobin is just kind of sitting here, ready for more oxygen. If you're a smoker, and your bloodstream is full of 2,3-BPG, what happens is it comes on, comes off, but a new one comes in. You're going to have more of your hemoglobin that's going to be bound with 2,3-BPG than a non-smoker. You're going to have a greater percentage of your hemoglobin when it gets back to the lung that's going to be stuck in the T state, unable to bind oxygen. This is why you as a smoker are going to be gasping for breath as you try to exercise. You have a lower oxygen carrying capacity because more of your hemoglobin is bound to 2,3-BPG and locking it in the T state where it's not binding to oxygen. Okay? So, we're going to talk in this term several things about effects of smokers. This is one of the biggest ones right here. Lainey?
Is it because you have to have oxygen being created?
Well, oxygen's in your lungs. It's not being created, but the hemoglobin that gets to the lungs isn't in a form that's going to bind oxygen very readily. So smokers, therefore, have a smaller percentage of their hemoglobin capable of carrying oxygen. So therefore, they have a lower oxygen carrying capacity. Yes?
So do smokers sometimes end up just making more hemoglobin?
Do smokers sometimes end up making more hemoglobin? That's a good question. I don't know the answer to that. You would think that they might, but I honestly don't know the answer to the question. Yeah. I would argue probably not, but because based on the breath thing that you see, because that doesn't go away. But I don't know the answer to that question. Yeah, Laurie.
So if 2,3-BPG doesn't get broken down and it's deoxygenated hemoglobin, it can't bind, or it cannot bind oxygen.
It's stuck in the T state. It doesn't bind it very well at all. I won't say it doesn't bind it at all, but it's a much lower affinity, much lower affinity. So the net effect is you have smokers have a smaller percentage of their hemoglobin capable of binding oxygen with high affinity. Okay, so hemoglobin's interesting that way. Now, there's the T to R transition. We don't need to talk about that. You've already talked about. I talked about what T to R is. All right, and it's not appropriate to talk about catalysis because this isn't a catalyst. By the way, I want to emphasize people: hemoglobin is not an enzyme, despite what your book would like to tell you. Okay, it doesn't catalyze any reactions. It simply carries oxygen. So even though it talks about catalysis and so forth, there's nothing being catalyzed here. There's no reaction that's occurring. It's simply carrying oxygen. But the scheme that they show is relevant. We'll talk about these schemes later when we talk about enzymes. Hemoglobin has, the scheme we see for hemoglobin here is a model of change of protein structure that is referred to as sequential. The binding of the oxygen by the first one caused that first one to interact differently with others. So you notice that we went from 4 squares to a circle and 3 rounded squares, or 2 rounded squares in this case. We've changed the configurations of those slightly. Those interactions have changed that protein. This, in turn, favors binding of another, favors binding of another, favors binding of another. These sequentially go through changes as a result of more and more oxygen binding. Okay? There's another model, I'm not going to talk much about it here, I'll talk about it next week, called concerted. And the concerted model says that the change, the flip occurs not as a result of binding of one, followed by another, followed by another, followed by another, but rather from a complete T to a complete R randomly. And that's not relevant for our purposes because hemoglobin follows this model pretty nicely. Yes, Cassandra.
Why is K4 more favored for someone on 3 oxygens rather than on 4? Does that make sense? The arrow.
Why is the arrow small here?
Yeah.
It's a little misleading. This actually depends on the oxygen concentration as well. So remember that I said that when we get to this state and we start dumping oxygens, that we can now favor the release, favor the release, favor the release. So all these really are variables depending upon oxygen concentration. So it's a good question, but it's, for our physiological purposes, not real relevant. Even at this thing right here in the lungs, there's plenty of oxygen to make this happen. But when you get out to the tissues, you want to start unloading, and that's really why that's happening. Make sense? Yes, Mike. Yes. Are there any hemoglobins that are only carrying 2 or 3? Yes, there are. So there's a mixture of things that are in there. Okay. Now I want to let's see. I wanted to say one. If you want to see the 2,3-BPG, that's its structure. Again, we're not worried about that. It's a small molecule. It's actually produced as a byproduct of sugar breakdown, as we shall see. Here's the effect. Okay. Look at what's happened. Here is, it's actually hemoglobin. They've sort of scrunched up this axis. It actually is an S-shaped curve. And, oh, by the way, I need to tell you about the shapes of the curves. The curve of binding for myoglobin that I showed you to begin with, it looks sort of like this, where it just went up and looped over. That's called a hyperbolic binding curve. And we see hyperbolic binding curves when we don't have cooperativity. So myoglobin doesn't have cooperativity because it doesn't have multiple subunits and it has a hyperbolic binding curve. The sort of S shape that we saw of that hemoglobin binding curve is very characteristic of cooperativity. And it's called sigmoidal. Sigmoidal. They flattened this, although these really are sigmoidal. They're trying to emphasize the difference in the binding affinities. And what they're trying to show you here is that when 2,3-BPG is present, hemoglobin has less oxygen binding affinity than hemoglobin without it. What this means is that hemoglobin, okay, when it doesn't have 2,3-BPG, is going to hold on to its oxygen more than when 2,3-BPG is present. Now, I know I hear the papers rustling. I've got one minute to make my point, and then I'll shut up. When we compare maternal hemoglobin, mom's hemoglobin, to fetal hemoglobin, there are some slight changes in the proteins. Fetal hemoglobin cannot bind 2,3-BPG. Now, I'll say something about that at the beginning of next period, but I want you between now and then to think about what that means in terms of not being able to bind 2,3-BPG for the fetus. Okay? That's the thought for them. See you guys on Friday.